My name is André Fenton. I'm a professor of neuroscience at New York University. I also co-direct the Neural Systems and Behavior course at Marine Biological Laboratories in Woods Hole. And I founded a company called Biosignal Group, and we make neurological devices that help patients and physicians manage neurological emergencies. This is part 2 of a series of 3 lectures that I will talk about how we think about memory and understand the neurobiology that underlies this feature of the human mind. I want to remind you that the work I'm going to talk about, which focuses on research that's been done in my laboratory, has been done by a very talented team of people, not only people in my laboratory, but people in other laboratories, and this work extends across decades. In part 1 of this series, I talked about how brains encode information in the activity of the individual neurons. And there are approximately 100 billion or so neurons in a human brain. And each of those neurons is able to communicate with approximately 10,000 or so other neurons. So that's trillions of communications, or the potential for trillions of interactions within the human brain, within the neural network there. And so that's not something that we're used to thinking about. And what I want to offer you is a conceptual framework for thinking about how information might be communicated and flow through the networks that make up your brain, the networks of neurons. And the internet provides a useful conceptual framework. In fact, this is an image of the activity in the internet. And what you can see here, if you think about the internet, it's just a bunch of computers that operate under a set of protocols or rules, and they exchange information. And in this image, you can see that some of the nodes in the internet, some of the computers or addresses in the internet, are very bright and have a dense number of connections. That means that many computers are trying to communicate or effectively communicating with those bright nodes. We think of them as hubs. These are the Googles and the Yahoos of the world, and the other companies like Akamai that we usually don't hear about that process a lot of the information traffic in the internet. And so if you think about that, what's interesting about the internet is it's self-organized. It's just a bunch of computers following some rules, and based on usage, some of those computers or addresses become more important than others, and they compete with each other for communication, if you will. And as more computers get used, more resources go to those computers or to those addresses so that they can store more information and process more information, and they can outcompete other computer networks or other hubs. And so the computer and their interactions are dynamic on the internet. It's never static in time, and it evolves. And in many ways, this is a useful way to think about how the activity in your brain evolves and is organized. With use, certain neurons change and collect more resources, outcompete other neurons, so that the functional architecture of a brain changes as that brain is used. So if you think about that, a natural question emerges. What guides and controls the information in the brain? Well, I ask you, what guides and controls the flow of traffic in the internet? You might be more familiar with traffic on the street, and the analogy applies. We could ask the same question. What guides and controls the flow of traffic on the streets? And this is analogous to the information communication within networks of neurons. So as traffic flows, you can imagine these as the action potentials or the discharge of neurons, single neurons, coursing through a network, not of streets, but a network of neurons in the brain. And what should be apparent is that this flow is not random. It's not random here in the traffic either. It's controlled in large part by the junctions. by how the different intersections are coordinated, and their coordination, we don't see it here, but their coordination is accomplished typically by traffic lights. And if you think about those traffic lights, what they're really doing is making it easier or harder for traffic to flow in a particular direction. And we will talk about the junctions between neurons called synapses, which can be modified to make it easier or harder for one neuron to communicate with another and to make pathways that facilitate and impede the flow of neural information through a network of neurons. These synapses, cartooned here, are the junctions of one neuron communicating with another neuron. Now remember, a single neuron on average in the brain would have 10,000 or so of these junctions. One neuron would connect to approximately 10,000 or so other neurons at each of these junctions. What's important about the junctions is that they allow an exchange of chemicals, shown here as the little green balls. These are neurotransmitters that can be released from what we call the presynaptic neuron onto the postsynaptic neuron. And the postsynaptic neuron has proteins, what we would call receptors, that upon receiving the neurotransmitter change the activity in the postsynaptic cell and can make that cell more or less likely to move, to use the analogy of traffic, and specifically more or less likely to generate an electrical discharge. And so what's important to recognize is these different interactions can be regulated. And what I'm going to now describe for you is some experiments that we've done in our laboratory that shows that the experience, in particular cognitive experience, the manipulation of knowledge, leaves a substantial detectable imprint, if you will, okay, or set of changes in the neural system that we'll study, okay, within the synaptic organization of the brain, and we'll focus on the hippocampus, which is a hub for storing memories. And this is the work that's been done in my laboratory as well as my collaborator, Todd Sachter, and in particular, really truly heroic work by Dr. Panos Tsokas. So the hippocampus, this might be a part of the brain that you've heard about because it's crucial. It's in the analogy, it's a hub for not only forming and storing, but processing memory. And so here's a animation that will help you orient as to where the hippocampus is. This is a mouse, and you can imagine slicing through the mouse's brain. And this structure in the middle that's delineated by the dark cells there, right, on one side and the other, that's the hippocampus. Okay? And that's the part of the brain that we're going to study. And we're going to study and talk about the neurons and the synapses in this part, in this region of the brain. We're going to need the animals or our subjects to actually form memories and communicate that they form those memories to us. And so this is a task that we're going to use called the active place avoidance task. You can see here a mouse on a grid floor. And the mouse, you'll notice, is staying down near, if you will, 6 o'clock or so of this, if it were a clock face, and it's staying away from the area that's been highlighted red. Now the area that's highlighted red is highlighted red for you and me. The mouse doesn't see red. What the mouse can do is look around and see where it is in the room and recognize that if it goes into the area that's been indicated by red for you and me, the floor will become electrified. And that's why the mouse is spending all of its time away from that area. So it's a rather simple task for the animal to learn to stay out of that red zone. And it does so by being active and walking away. And that's why we call it the active place avoidance task. And while the mouse can't speak to us and say, oh yeah, I remember I was shocked over there, what it can show us through its behavior is that it's, for some reason, staying away from that area. And the natural inference would be, because it has remembered that it was shocked in that area. So we can use this task to assess and to quantify and measure the extent to which the mouse remembers where it was shocked. And while doing this task, what we can do is record neuronal activity, okay, shown here in cartoon form. Imagine 15 cells and each tick corresponding to the discharge, the electrical discharge of a single neuron in the hippocampus. What's shown down below in green is a slice through the hippocampus of a mouse after it has learned this task, and this mouse has been genetically engineered such that the neurons that were active in a strong manner while the animal was learning this task are now green. And so we can recognize, and there are a couple of things to notice. Not all cells are green. Only about 30 or maybe even fewer % of the cells are green. And the allocation of the green cells is not homogeneous in this structure. We won't talk about the anatomical confines, but you can notice that some places the green cells are more dense than others. So different regions of the hippocampus as a neural circuit activate and allocate different numbers of cells to form the memories that this mouse is forming and going to use in order to organize where it goes in space and presumably remember where the shock was. But we might now ask, that was the easy question to answer, right? We might now ask, okay, how do the neurons remember whether or not they should be active? And in order to study that, okay, what we understand is that the synapses, the connections between the neurons, these are the things that actually differ and they can change with experience. That's the dominant hypothesis for answering the question, how do the neurons remember whether or not to be active for one memory or another memory? And now let's talk a little bit about the neurobiology and some of the findings that support that point of view. This story begins with an important paper by my colleague, Todd Sachter, where he identified a particular molecule called Protein Kinase M zeta, PKM zeta. And what he was able to show is that it's necessary and sufficient for something called LTP maintenance. And what's cartooned here is if you were able to make a measurement of the electrical activity of a single one of these neurons in the hippocampus, what you could see is and measure is how easy is it for that cell to generate an electrical current. And what we see here shown on the x-axis is time, and on the y-axis is shown the amount of current that could be measured from across a pair of these cells. And what you can see is that in one of them, starting at time 0, the current is going up and then it stabilizes. It takes about 10 minutes to go up and then it stabilizes. And the trace that shows the increasing current, what the folks in Todd's lab did was they delivered into the cell, okay, protein kinase M zeta. So it was sufficient to increase the electrical communication between neurons. And other experiments, I'll show you one now, that I did with Todd, shows that it's necessary to maintain this enhanced electrical activity. So here's an experiment, truly heroic experiment done by Eva Pastolkova when she was in my laboratory. What Eva did was she gave a high frequency stimulation at time 0, and that caused neurons, many neurons, thousands of them, to discharge together, and that active discharge together caused them to subsequently increase their response to a simple stimulation. So they became elevated, if you will, or enhanced, and what we say is that their response was potentiated, and it was potentiated for a long period of time. In fact, in Eva's experiment, the potentiation lasted at least 24 hours and would have probably gone on, except that what Eva did was she injected an inhibitor, okay, of this molecule protein kinase M zeta. And you can see that the ZIP stands for zeta inhibitory peptide. And what you can see is when ZIP was injected, okay, that the potentiated response went back down to the baseline. And so that's quite remarkable. There are no other known molecules that one can say are both sufficient as well as necessary for maintaining the activity-dependent enhancement of communication between neurons. And so we decided to study whether or not PKM-zeta was also important, whether or not it was crucial for maintaining memories. The first thing to note, okay, is if you give an animal, or like a mouse or a rat, the training in the place avoidance tasks that I showed you, okay, you can actually find that if you look 1 day later or 1 month later, by grinding up the brain and measuring the protein level of PKM-zeta, what's shown here in the Western blots, or some control protein, actin, what you can see is after 1 day or even after 1 month after the training, the levels of PKM-zeta have elevated. And that's really quite remarkable because, again, I'm unaware of any other molecule that, based simply on experience, will have an elevated expression like that. Certainly no other memory molecule, again, I'm aware of. And so that's quite enticing. Experience itself is sufficient to increase the activity, and it does so in a persistent way, lasting at least 1 month. Okay. And so where does PKM-zeta reside? PKM-zeta resides on the postsynaptic side of the synapse. Okay. And it's a molecule that engages and modifies other molecules that change the expression of the electrical response when one cell is active, when the presynaptic cell is active and releases its neurotransmitter to the postsynaptic cell. And if we were to look and make slices, instead of grinding up the brain, if we make slices and stain the brain so that we can find out where is the PKM-zeta, we can see that after 1 month, in an animal that received this training, so 1 month later we can sacrifice the animal and look for PKM-zeta, and we see the hotter colors, if you will, on the right side in the trained animal compared to the control animal. And the control animal had exactly the same experience. The shock was simply never turned on. Okay, so PKM-zeta seems to form some kind of a long-lasting trace, if you will, of the experience of the animal. And we would imagine and work under the hypothesis that that's a trace of the experience, the kind of thing that we call a long-term memory, and how it's maintained in the brain. So to actually test that idea, it's not sufficient simply to look and see that the molecule is there. We actually want to make a manipulation. And so this is a video I'll show you of an experiment that we did. And what we did in this case was we trained in this experiment, it was a rat. We trained rats on this rotating arena to walk around first of all. Right? In this case, the shock isn't turned on, the rat is walking around, the rotation is about 5 times faster, otherwise it would be very boring to watch. You'll notice that the rat goes everywhere, and we can track where the animal goes, and the computer can decide, based on where the rat is, whether or not to electrify the floor. And so we have training trials where the floor is electrified when the animal is detected in a particular part of the space, a particular part of the room. And it can know where it is in the room by looking at the cues that are on the curtain and such that aren't necessarily visible to you. The red mark on the arena is there just so that we can see what the computer has decided to shock. So now we can do the experiment. we can do a control injection just of saline into the hippocampus a day later and put the animal back and watch, does it express memory by staying away from that place that is shocked. And if it does, as in this case, you'll see that it runs away, it just enters and runs away. We didn't turn the shock on. Presumably it ran away because it remembered the location of shock. But what if we inject an inhibitor of PKM-zeta? What we see instead is the animal doesn't avoid any longer. It seems to walk around as if, in fact, it was naive for being in this arena. And in fact, as if it was naive, certainly, that the shock was ever delivered in the upper part of the arena. In fact, if we compare that same rat, its first time in the arena with the time after the zip injection, it's very difficult to tell the difference. Okay? And this makes it look like it's possible for the first time to erase a memory, because what we did in subsequent experiments is show that after this erasure, if you will, the animal had no difficulty to form a new memory. Okay? It could form short-term memories, and it could even form long-term memories after the zip had been metabolized and removed a couple of hours later. So we hadn't damaged the brain in any way. And because it looked like it was now possible to edit, if you will, to erase memories, whether or not specific, this got the attention of quite a number of people in the public media, as well as our friends in the scientific world. In fact, that inspired some other researchers to test that idea. And they had a very interesting and reasonable approach. They said, well, if PKM-zeta is so important, if it's crucial for memory, then it should be possible to genetically delete PKM-zeta in a mouse. And if we did that, then the mouse should not show normal learning, should not be able to acquire memories, it should not have the ability to remember, and it should not have the ability to make LTP, this long-term potentiation of the synaptic transmission between neurons. But as you can see from the titles of these papers, right, deleting or knocking out the gene for PKM-zeta and the mice look pretty normal. And they remembered rather well. So that led these authors to conclude that PKM-zeta was not crucial for long-term memory or LTP, and that in fact our inhibitor must have been acting on something else. So this is the nature of science, and what is interesting about science is that it breeds controversy. And with controversy, it breeds further investigation. Very recently, we explained how it is possible that by deleting the gene, you could still observe that there would be normal learning and memory when, in fact, that gene is indeed crucial for learning and memory. And I'll explain now in the next few slides. The answer was quite simple. In part one, I talked about how we need to think about complex systems like the brain and complex processes like memory at multiple levels of biological organization. And the answer was simple. There was compensation. When the gene for PKM-zeta was deleted, an alternative gene, another related, in fact, very closely related gene, itself became expressed. And otherwise, in a normal or in a wild-type animal, its expression tends to be low or suppressed, presumably by the presence and actions of PKM-zeta in memory. And so what you can see here is that this other protein called PKC iota lambda, they're part of the same family of proteins called protein kinases. And if you look on the left, you can see that in a knockout mouse, okay, when you take the half brain, the hemibrain of that mouse, and you measure how much PKC iota lambda is there, okay, you can see that it's about the same. But if you look in the specific part of the brain, the hippocampus, that's crucial for memories, there's an overexpression by almost 60% of that particular molecule. And this is just spontaneously. And in fact, if you look at the active form, which is a phosphorylated form of the molecule, you see that the increase is over 400%. So there was a massive increase of this other molecule, PKC iota lambda. But that doesn't prove there's compensation. That just proves that there's a suspect. So we had to do more work. The logic is, if PKM-zeta is crucial for memory in wild-type animals, but is compensated, and therefore the mechanism for memory and the mechanism for LTP is different in the PKM-zeta knockout mice, then we should be able to selectively inhibit the new synthesis of PKM-zeta to prevent LTP, and I'll show you subsequently memory, but only in the wild-type mice. So you can look here at this trace. Along the x-axis is time, and on the y-axis is a measure of the synaptic strength or the synaptic response to stimulation of presynaptic cells and how the postsynaptic cells are responding. And at time 0, a strong stimulation is given to induce this long-term potentiation. And if you look at the wild-type mice in green that received a scrambled version of this inhibiting molecule, we used an antisense oligodeoxynucleotide, which is something that will bind to the DNA that's designed to interfere with the manufacture or expression of a new PKM-zeta molecule. When we inject that, what we can see, or when we put that in the bath for measuring LTP, what we can see is in the green trace, when that oligodeoxynucleotide is a scrambled ineffective form, we see normal LTP. But in the red, you can see that the LTP is able to be induced, but it doesn't sustain. Okay? And it seems to have no effect in the null. So that's the first, if you will, piece of evidence that the mechanism for LTP is PKM-zeta dependent in wild-type mice, but different in the knockout mice. The second is the complementary experiment. You can now try to inhibit the PKC iota lambda. And there was a specific inhibitor for that called ICAP. And you can see there, if you give ICAP in the upper trace, to established LTP, when you add the ICAP, the LTP goes back towards baseline, it depotentiates, but only in the PKM-zeta null mouse that's not making the PKM-zeta protein. Whereas in the wild-type mouse, right, which can make PKC iota lambda as well as PKM-zeta, this inhibitor has no effect on the maintenance. That's the black trace. If you look at the open circles on the bottom, you can see that second arrow at time 270 minutes. That's a second stimulation that we can give. And what you see is that it becomes difficult to establish the LTP. And so PKC iota lambda has been known to be important for the initiation of LTP and memories, but it seems to not be crucial for the maintenance of it, at least in the wild-type animals. But it seems to act as a backup mechanism when you get rid of the PKM-zeta through the genetic deletion. What does this do for long-term memory? So what we can do is repeat these experiments. We can use the antisense, okay, and we can deliver it before every memory trial. And what's shown here in these circles with the traces of where the mouse has gone, okay, after PKM-zeta inhibitor, the antisense has been injected. And we can see in the pre-training, the animals walk around everywhere. In the training sessions, they walk around and they can learn and acquire this information during the 10 minutes. Remember, PKM-zeta is crucial for the maintenance, the maintaining of the memory, not its acquisition. Okay. And we can test retention in this case 1 day later, and you can see in the wild type traces the first two, the scrambled or ineffective peptide doesn't seem to impair the animal's ability to remember. In the wild type, the antisense seems to create that kind of impairment. Now the animal enters the shock zone very quickly. The trace is very short before it enters the shock zone. The scrambled peptide or the antisense seems to not have an effect. You see a large trace there. The animal spends a lot of time before it first enters the shock zone in the PKM-zeta knockout or null mouse. And this can be quantified in the summary data. You can see that after delivering the scrambled peptide to the wild-type mice in the green bar, that the retention, the time it takes to first enter the shock zone is long. However, not after delivering the antisense to prevent the synthesis of PKM-zeta. And in the PKM-zeta null mice, these things don't have an effect. Those gray bars are quite tall, indicating good memory, as I showed you in the pictures in the previous panel. What about the PKC iota lambda inhibitor, ICAP? So now what we can do is train the animals to establish a long-lasting memory, both the wild type and the PKM-zeta null mice. If you look at training trial 3, they both established a memory. They're staying away from the shock zone. And now, 1 day later, before putting them back in the arena, we can deliver the PKC iota lambda inhibitor, and it seems to have no effect in the wild-type mouse. It continues to stay away from the shock zone. In fact, in this picture, it doesn't enter at all. Whereas in the PKM-zeta knockout mouse, or the null, it goes everywhere and is acting as if it is naive. And that's also shown here in the summary data. The dark bar in the wild-type mouse showing the memory or the measure of how long it took to enter the shock zone is very high in the wild-type mouse, but in the null, it's very short because it doesn't demonstrate memory. Again, evidence that the mechanisms of maintaining memory are distinct in the wild type and the PKM-zeta null mouse. In fact, what we can look at is the detailed behavior in the PKM-zeta null mouse and ask, did they actually learn in a normal way? And if you look at the details of where these animals actually spend their time, you can see in the wild-type mouse, early in training, if we give them a training protocol that's harder to acquire, early in training, they spend their time, see the red activity, that's the density of where the animal's spending its time, is across from the shock zone around 6 o'clock. And it happens to be there right on day 1, and it in fact intensifies with training. But look in the PKM-zeta null mouse, right? That red area is closer, if you will, to about 9 o'clock. And it only very gradually, it takes 7 to 10 trials before it actually moves. The animals decide to spend their time in the safer part of the arena. And if you look and quantify this, you can see it takes the animals, you can measure the ratio of time spent opposite the shock zone, which is the safer part of the shock zone, versus the safer part of the arena versus the part adjacent to the shock zone. And what you can see here is that ratio is declining gradually in the wild-type mouse, and it's established already within the first day. However, it takes some time for the PKM-zeta null mice, about 7 or so trials, which means 3 days of training before that drops down. And you can also see by measuring the strength of the memory as the time it takes for the animals to first enter the shock zone, it takes much longer for the open circles, the PKM-zeta null mice. They take much longer to get up to their asymptote, to the level of performance, and that level of performance is about half of that as the wild-type mouse. So their memory seems to be abnormal, and their way of acquiring information also seems to be abnormal in the PKM-zeta null mouse. Again, they are different than the wild-type mice. And we can look at a different memory task. In this case, what we can do is have animals walk around in 3 different boxes. Cartooned here is green, yellow, and blue, but they just look different and smell different to the animals. And what we can do is take advantage of the ability or interest of mice in different objects, and they respond to novelty. And so the animals were made familiar with different numbers of objects in these boxes, and in the one case what we can do is after they've been familiar for a day and stored presumably in memory for those objects, what we can do is exchange the objects, for example, from context B, those orange objects, we can exchange them for a pair of the objects in context A and put the animal back and look to see where the animals spend their time. Do they investigate the novel objects? And they do. Just like the wild-type animals. And we can also ask, what happens if we just exchange a pair of the objects, okay, that were already in the context B? And so you can see the circle has been exchanged for the cube in the yellow case. And this is a much more subtle distinction. You can think of the first distinction as if, for example, someone were to exchange your toothbrush for the fork at the dinner table. That would be something that might gather your attention. But much more subtle is exchanging the fork, for example, with the spoon. And that subtle distinction the animals don't seem to remember if they're missing the PKM-zeta molecule, as in the case of the PKM-zeta-null mouse. And if you simply exchange the locations of a pair of objects, they have, again, very strong memory for that and no difficulty in recollecting that. So what I've shown you so far is that PKM-zeta is crucial for the persistence of long-term memory. Okay? And you might ask another question, right? At least it's crucial for the persistence of long-term memory in normal or wild-type mice. Okay? But we might ask another question. Does PKM-zeta actually increase, okay, in long-term memory? If it's maintaining the memory, how does it do that? A simple, the most parsimonious way to accomplish that would be to make more of it. And so I'll show you some experiments. We can use the same experimental task. We can train animals to form a memory. And in this case, we can train them to form a one-day memory. In the place avoidance task here, you can see the control animals, the shock was never turned on, they're just walking around, whereas the trained animals, they stay away from the shock zone, and when we measure how long it takes them to enter the shock zone, it's elevated if you look at the dark bars a day later. And a day later, we can see that where the asterisks are, that the amount of PKM-zeta studied by Western blot is enhanced one day if the animals have had spatial training, whether or not they actually were tested for their memory. The elevation already happens 2.5 hours after the training, but it doesn't happen within 5 minutes if the animals have, if you will, only established a short-term memory. So it takes some time for PKM-zeta to get made and to be elevated, and then it can remain elevated persistently. The control experiment here is a group of animals received an unavoidable shock. So every time the trained animal was shocked, this group of animals was also shocked, and that doesn't itself increase the PKM-zeta. It's really necessary to form a memory for the zeta increase. And that, in fact, if you were to look now at just the data from the animals that had the spatial training on day 1, and you were to measure how well they remember, shown on the y-axis, their time to first enter, and you looked at the amount of PKM-zeta that was expressed in their hippocampus in the x-axis, you see that there's a strong relationship. Those animals that expressed very low levels of PKM-zeta in their memory retention show weak memory retention. Those animals that express very strong levels of PKM-zeta after memory retention show strong memory retention. So it seems that PKM-zeta both increases with and is correlated with the expression of long-term memory. So how long does this increase last? We can repeat this experiment, okay, and instead of testing 1 day later, we can test 1 month later. Okay. What you can see here is with this particular training, the animals show very poor memory recall 1 month later. They're going everywhere. And when you measure PKM zeta levels, you can see that the PKM zeta levels in green are no different from baseline. They're not increased a month later when the animals show no memory 1 month later. The bars on the graph show the control animals who never formed a memory, and the animals that were trained but 1 month later also don't show memory expression. And PKM zeta also isn't increased by Western blot. But if we train the animals a little bit differently so that they form a 1 month memory, as shown here, you can see now the trained animals are staying away from the shock zone at the retention test given 1 month later. The gray bar shows that it's elevated. They have an elevated time to first enter. Well, you can also see that in the green bars, the trained animals have now an elevated PKM zeta level. And this is now 1 month after the initial training. So PKM zeta seems to have a persistent increased expression with long-term memory that lasts at least a month. Okay? And that's quite remarkable. And I remind you that when we looked 1 month later, we could also see where PKM zeta was distributed in the hippocampus, and that it's not everywhere in the brain. It's not even everywhere in the hippocampus. It's in particular subregions of the hippocampus. The subregions, in fact, that we can study the electrical activity and the changes in how the conjunction of electrical activity is able to manifest as increased synaptic potentiation, which is what we, remember, hypothesize as a field is the underlying expression or cellular mechanism for storage of memory. So to summarize, what we've talked about is the molecular mechanisms that underlie memory. We don't know them yet. There's active controversy, okay, that I just described. Okay? But we have strong hypotheses, and we're well on our way to delineating what the molecular neurobiological mechanisms are that sustain memories. PKM zeta is a molecule that seems to be crucial for the persistence of long-term memory. Remember, it increases while animals hold or form memories, and it seems to persist as long as long-term memories persist. When the long-term memories decline, we see that PKM zeta increases, decline back to baseline levels. And so we would conclude that PKM zeta is at least the leading candidate component, right, of the molecular mechanism of memory maintenance, which is actually saying something substantial in the effort to understand the fundamental neurobiological underpinnings of something as crucial and important to our existence as memory. And we've been able to trace, by understanding and studying the brain at the level of behavior, at the level of neural circuits, at the level of physiology, and the level of gene expression, the molecular constituents of the mechanism for the storage and persistence of memory. And I'd like to thank the large number of people that have contributed to this work, both in my laboratory as well as in collaborating laboratories, and of course, the generous funding that has made all of this research possible across the decades.